Welcome everyone to today's presentation as part of the Advance-CTR Clinical and Translational Seminars series, which is held monthly. For those of you who don't know me, my name is Ed Hallrott, professor at Brown University, and I have served as the program coordinator for Advance-CTR. And I will be hosting Dr. Van Nostrand today. Our fearless leader and PI, Sharon Rounds, is out of town and has asked me to fill in for her. So let me say that we very much welcome our colleague from URI, who is the co-executive director of the George and Ryan Institute for Neuroscience and a professor in the Department of Biomedical and Pharmaceutical Science at the University of Rhode Island. He came to us in Rhode Island in 2017 from the Department of Neurosurgery at Stony Brook University. And he has been working in the field of Alzheimer's disease and related disorders for over 30 years and is one of the field's leading researchers on the pathogenesis of cerebral amyloid angiopathy and a cerebral small vessel disease and Alzheimer's disease. He's been trained as a biochemist and was very much involved in the first purification and characterization of the now infamous amyloid precursor protein, the progenitor of amyloid beta, and a key component involved in the pathogenesis of Alzheimer's disease. We very much look forward to the seminar, which is entitled Molecular Insights into the Pathogenesis of Cerebral Amyloid Angiopathy Using Novel Rat Models. Please take it away.
Yeah, well, thank you very much for a nice introduction, and thank you all for joining me here for this presentation this afternoon. So I'll just get started here. First, I just want to state my disclosures. In the past, I've received some research funding from L&M Pharmaceuticals, and I currently receive some research funding from Eli Lilly. So for today's presentation, I just like to give a little outline here about how I'll be breaking up the talk. First, especially for those who may not be all that familiar with CAA, I want to give some background about vascular cognitive impairment and dementia, discuss a bit about the amyloid beta peptide, and then about cerebral amyloid angiopathy, the topic of the talk today. I would then like to move on to describe some of our work where we've generated and characterized transgenic rats to model CA type 1 and CA type 2. And for the final part of my talk, I'll be discussing some of the proteomic analysis we've done in these rat models to gain insight into the molecular pathogenesis of the disease. So first, I talk about this term vascular cognitive impairment and dementia. And this was coined a number of years ago and was used to describe a form of dementia that is triggered by damage to cerebral blood vessels or cerebrovascular disease. And there are a number of conditions that can cause VCID. I have a number of those listed here. They can include hypertension, cerebral hypoperfusion, arteriosclerosis, hyperhomocystinemia, diabetes, neurovascular inflammation, and ischemic stroke and hemorrhagic stroke. And the other condition is cerebral amyloid angiopathy, what I'll be speaking about today. So before I get into that, and we'll be talking about the amyloid beta peptide, I want to talk a little bit about the generation of that. So we are all quite familiar with the amyloid beta peptide and its relationship to Alzheimer's disease and related disorders. So this again is a small peptide that's derived from the amyloid precursor protein, which is a diagram shown here. This is a large type 1 integral membrane protein with a large extracellular region, a single membrane spanning region, and a small cytoplasmic C-terminal tail. The A-beta residues of the amyloid precursor protein are highlighted here in pink. So it's been well worked out now, the proteolytic processing of the precursor that gives rise to the amyloid beta peptide. Initially, there's a cleavage at the end terminus of the A-beta peptide sequence by an enzyme termed beta secretase. This releases the large extracellular fragment into the extracellular environment, and the intact amyloid beta protein remains intact with the membrane spanning region and the C-terminal portion of the protein. That subsequently then goes through a cleavage process by gamma secretase, which involves presenilin proteins, and this releases the amyloid beta peptide. This processing generates primarily two forms of the amyloid beta peptide. The most prominent form that's generated is A beta 40. That accounts for greater than 90% of A beta that's generated, and much less of A beta 42, which is less than 10%. A beta 42 contains these two extra hydrophobic amino acids here at the C-terminal region. And because of this, and also it's believed that this is the more pathogenic variety of the peptide because it has a stronger ability to self-assemble into beta sheet-containing amyloid fibrils. And below here, this is an electron micrograph of A beta 42 fibrils, what they look like when they're assembled. So to talk about CA, again, this is a very common pathology of the elderly. And there have been a number of studies that have estimated that more than 50% of individuals over the age of 80 have some level of CAA within their brains. And the fact that CAA involves the amyloid beta peptide, it's not surprising that it's the most common vascular pathology associated with Alzheimer's disease. This is a tissue section from an AD patient here, which was stained with a dye thioflavin S to visualize fibrillar amyloid deposits. And you can see here abundant parenchymal amyloid plaques within the neuropil of the brain. And concomitant with that, you see amyloid contained within the vessel wall, CAA. So this accumulation of amyloid within the cerebral vessel wall actually results in a number of pathological consequences. First, it can lead to disruption of cerebral blood flow. It can cause perivascular neuroinflammation. Importantly, it can disrupt brain waste clearance mechanisms through perivascular and paravascular routes. It can also cause cerebral infarcts, micro and macro hemorrhages. And all these conditions can contribute to vascular cognitive impairment and dementia that's seen in CA patients. Now, this pathology of CA has actually been in the spotlight more recently because it's been shown to increase detrimental amyloid-related imaging abnormality effects associated with AD immunotherapies, and most notably bleeds in the brain. So there are two forms of cerebral amyloid angiopathy. The most common form is known as CA type 2. Here's an example of that shown in a patient. In this case, the amyloid affects arteries and arterioles and the cortex and meninges. And it's shown here that the fibrillar amyloid is largely confined within the vessel wall, as you can see here with the staining with thioflavin S showing the fibrillar amyloid. And generally with CA type 2, there's a lack of a robust neuroinflammatory response in the absence of dysoric amyloid. So this term dysoric means without borders. In this case, in CA type 2, the amyloid is contained within the borders of the vessel wall and therefore does not promote, again, a strong inflammatory response. Now, the other type of CA is CA type 1. And here's an example here of a patient with CA type 1. In this case, it affects small microvessels and capillaries and can occur with or without larger vessels CA type 2. And in this case of CA type 1, the amyloid now actually penetrates into the surrounding brain parenchyma, as you can see here. And because of this, it promotes a very robust neuroinflammatory response because now there's a presence of the dysoric amyloid that's now exuding out beyond the borders of the vessel. So in addition to the cerebral amyloid angiopathy that occurs sporadically and is associated with Alzheimer's disease, a number of familial forms of CA have been identified. And these familial forms tend to result from mutations within the A beta peptide that generally cluster around the mid-region of A beta peptide. And a number of these are listed here. Probably the most recognized form of familial CA is known as Dutch type CAA. This results from a point mutation in the APP gene that results in a change in the codon coding for what normally is a glutamic acid residue at position 22. It is now replaced with the glutamine residue at position 22. Patients with this mutation develop early onset and severe CAA. Here's an example of a patient with Dutch type CAA, and you can see the extensive amount of amyloid accumulated within the vessel walls of these patients. Now, despite the fact that they accumulate very early onset and severe CAA, they do not develop parenchymal fibrillar amyloid plaques that are typically seen in Alzheimer's disease, nor do they develop neurofibrillary tangle pathology. However, these patients do develop recurrent and often fatal intracerebral hemorrhages at midlife, and they develop progressive vascular cognitive impairment and dementia. Now, another familial form is known as the Iowa type CAA. This again results from a point mutation in the APP gene. And again, this changes the codon that normally encodes for an aspartic acid residue at position 23, one site downstream from where the Dutch mutation occurs. In this case, it's replaced by an asparagine residue. Here's an example, again, of a patient that has the Iowa type CAA. Again, it's an early onset and severe form of disease. There is some larger vessel CA type 2, but you can see here there's also a very pronounced CA type 1. You see a lot of capillary involvement in this disorder. These patients develop micro-hemorrhages, but large intracerebral hemorrhages are less common. They do develop neurofibrillary tangled pathology, but not extensive parenchymal amyloid plaques like Alzheimer's. And they also develop a progressive form of vascular cognitive impairment and dementia. So we earlier spent a lot of time in my lab looking at some of the biochemical properties of these mutant forms of A-beta to understand how they're associated with disease. And one of the things that's been noted from our earlier work and has been shown by other laboratories as well is that the presence of these mutations in A-beta associated with familial forms of CAA greatly enhance the fibrillogenic properties of the peptide. So here I've shown, what I show here is a thioflavin T progression curve. And what happens is, what we do is we resuspend A-beta peptide in solution in the presence of a dye, thioflavin T. As A-beta assembles into amyloid fibrils, it binds the thioflavin T dye and emits a fluorescent signal that we can read here, which is shown here. And that's, again, reflective of amyloid fibril formation. So if I draw your attention to the black line here, this is the non-mutated wild-type A-beta 40 peptide. And under these conditions, you can see it's a very poor assembler into amyloid fibrils. However, in the presence of the Iowa mutation in blue or the Dutch mutation in green, you can see this lag time is dramatically reduced and you see rapid formation of amyloid fibrils. Now, during the course of these studies, we generated a chimeric form of a mutant A-beta that harbored both the Dutch and the Iowa mutation. And what we found is that this is even more fibrillogenic. You can see there's a very short lag time that rapidly assembles into fibrils, much more rapidly than either the Dutch or Iowa mutation alone. We find similar effects when we look at cytotoxicity of these different forms of A-beta. This here is an in vitro model that we used of CAA early in my laboratory. These are cultured human cerebrovascular smooth muscle cells. They're treated with these different forms of A-beta. You can see that A-beta 40 wild type with no mutation, which again is a very poor assembler into amyloid fibrils, is not toxic to these cells. However, if the Dutch mutation or the Iowa mutation is present, we see enhanced toxicity. And notably, if we look at the chimeric Dutch Iowa mutant form of A-beta, again, which rapidly forms fibrils, it's even much more cytotoxic than either single mutation alone. So we have characterized these properties, again, looking at fibrillogenesis and toxicity, and to understand how these mutations may actually affect the physical nature of the amyloid beta peptide. So here, I'd just like to briefly just show some work that we published last year in collaboration with Steve Smith's group down at Stony Brook University. These are cryo-EM structures that were derived from vascular amyloid fibrils that were seeded from sporadic CAA. And his group, they had a resolution down to 2.9 angstroms, which is quite good. And here is just a molecular model of showing two A-beta subunits interacting in this fibrillar structure. And below here is the same model here, but just identifying the amino acids here. And these are two A-beta subunits here, and each one of these is a beta hairpin. Here, the N-terminus going to the C-terminus. Here, the N-terminus going to the C-terminus. What these studies showed is that there is a highly structured N-terminal region of A-beta within this fibril structure. And also, you see there's a strong hydrophobic interaction between each A-beta subunit. Again, it's shown here. One finding that came out of the study is, like I like to point to here, is actually at the N-terminus, and this is an electrostatic cluster. So that's actually blown up and shown here. And through these studies showed that there's an electrostatic interaction between the free amino group of aspartate number 1 at the N-terminus of A beta, this amino group interacts with the carboxyl side chain of glutamic acid at position 22. Similarly, there's an electrostatic interaction between the aspartic side chain of aspartic, the carboxyl side chain of aspartic residue number 23 with the epsilon amino group of lysine 28. So we have these electrostatic interactions here, which actually hold the A beta peptide into a tight structure. So what this actually identified, here again is that structure just for reference up here, of why these mutations may actually affect A beta. So when you look in the case of the Dutch mutation, again, normally there's an aspartic acid residue there. We now have, a glutamic acid residue. We now have a glutamine there. So we lose this acidic side chain for a neutral amine, which disrupts this interaction. When you look at the Iowa mutation, a similar thing happens. Again, normally there's an aspartic side chain, a side chain here, a carboxyl side chain with the aspartic acid residue. That's now replaced with a neutral amine. That disrupts this interaction with this epsilon amino group here at lysine 28. So what we hypothesized is actually by disrupting these electrostatic interactions, it loosens the rigidity of the A beta subunit and gives it more flexibility. That flexibility enables it to more rapidly assemble into beta sheet containing amyloid fibrils. And this is consistent again with what we find here. We look at the fibrinogenesis curves, as I showed earlier. Again, when the structure is tightly held in place with these electrostatic interactions, we see that there's poor assembly of fibrils here. But when you disrupt them, either with the Dutch or Iowa mutations, you increase the flexibility and also you increase the rate of amyloid fibril formation. It also indicates why when you disrupt both these interactions, that further destabilizes this N-terminal region, giving more flexibility within that A beta peptide subunit, allowing it to more rapidly assemble into amyloid fibrils. So we think that this actually provides a molecular understanding as to why these mutations lead to enhanced fibrinogenesis. So that's just, again, some background about A beta and some of these different forms of CAA. And what I'd like to do now is to shift over into work that we have described, so the work that we've been generating and characterizing novel transgenic rats to model CA type 1 and CA type 2. So historically and currently, the ADRD field largely relies on the use of mice to model disease, and these are by and large transgenic mice. These mice have been in the field for over 20 years. People could and use them and continue to use them. Although they've been very informative, we felt that we needed to try and make some improvements to make models better and more faithful to human disease. So because of this, we switched from mice over to using rats as a platform for modeling CAA. So there are several advantageous reasons for using rats compared to mice. First, from an evolutionary standpoint, rats are much closer to humans than mice. Their brains are much larger than a mouse brain. And because they have the larger brains, they have a much larger amount of white matter that they want to investigate. It's also much easier to obtain large volumes of CSF compared to a mouse. Now, unlike mice, rats contain the full complement of tau isoforms like humans. And this is important because it's been shown that when rats develop fibrillar amyloid plaque pathology in their brains, in response to that, they develop hypophosphorylated tau pathology like you see in humans. And this happens endogenously. This is something that does not occur in mice. There are more neuroimaging capabilities available at rats, and there are more sophisticated cognitive testing available. So again, because of these advantages here, again, we've decided to use the rats as a platform to move forward with our newer models. So the first model I like to just describe to you today is our RTG-DI rat model of capillary CA type 1. Here, what we did is we designed these rats to express human APP in neurons, and that APP is harboring both the Dutch and the Iowa mutations, with the idea that these rats would produce this chimeric Dutch Iowa mutant A beta in their brains. The reason why we put both these mutations in is because I showed on the earlier work that this chimeric peptide forms fibrils much more rapidly and it's much more cytotoxic. So that's why we initially used this chimeric form. Here again is the amyloid beta peptide, and these are again the two substitutions, the glutamine for glutamate at 22 and the asparagine for aspartate at position 23. So this is the peptide that they produce in their brains. So what we characterized and showed is that this is a model actually that develops early onset and severe capillary CAA type 1. So these figures over here, this is at 3 months of age, looking at different brain regions, the cortex, hippocampus, and thalamic area. They're stained in red to identify the vessels, and the green is stained with thioflavin S to identify fibrillar amyloid deposits, which you can see appearing at 3 months of age. This progresses out to 12 months of age. It gets very severe. You can see here that much of the capillaries here are completely covered by vascular amyloid. That was quantified here. Again, showing at 3 months, we see the emergence of capillary CAA type 1. When we go at the 12 months, we see this increases dramatically and is very pronounced within the hippocampus and thalamic regions. So subsequent to that model, we generated another transgenic rat known as RTGD. In this case, we took a similar approach to what we did with the RTGDI rats. Again, we were expressing human APP in neurons here, and but difference to the other one, difference to the RTGDI, here we just included the single Dutch mutation. And the idea behind this again is that we would have a rat expressing human Dutch mutant A beta within their brains. So this again is the A beta peptide sequence. In this case, again, we're just now producing Dutch mutant A beta with the E22Q substitution. We showed that these rats actually develop cortical and thalamic as well as surface vessels. Here we have surface meningeal vessels and penetrating arterioles. And this is CA type 2, again, stained with thioflavin S. You can see that, again, strong accumulation of vascular amyloid in these larger vessels. That's quantified down here over time. Now, in contrast to the RTGDI rats, which have a very early onset, showing CA at 3 months of age and gets very severe at 12 months, this is a much slower developing model. The CA does not appear till after 12 months, but does progress out to 24 months. So just again to emphasize that we have these two different models, and there are two different types of CAA that affect two different vascular beds. This again here is the RTGDI, the model of capillary CAA type 1. Again, you can see extensive amounts of amyloid on the capillaries in these brain regions. We do not see much larger vessel involvement in this model. So in contrast, as I just showed you in the RTGD rats, on the previous slide, I showed you that they develop primarily larger vessel CAA type 2. When you look at the capillary beds of these rats, they do not accumulate amyloid at all. So we have two very distinct models here, one of CAA type 1 and one of CAA type 2. So we went and characterized these the pathology in these models. First, we looked at the composition of the amyloid that accumulates in the vessels in these different models. So here's RTGDI. In the top panels here, they were immunolabeled with antibodies either specific to A beta 40 or to A beta 42. You can see that's predominantly A beta 40 that's labeling. We also went and isolated capillaries from these rats, shown here. You can see the amyloid deposits on them. We then extracted the amyloid from these capillaries. Here, these are the amyloid deposits from these capillaries. And we performed a ELISA analysis and showing that again, 90 to 95% of the A beta that's accumulating on these capillaries is indeed A beta 40, the shorter life peptide. We found a similar thing looking in the RTGD rats. Here again is an immunolabeling with antibody specific to A beta 40 and A beta 42. Again, you see very strong labeling with A beta 40. In this case, we isolated the larger vessels here. You can see these vessels full of amyloid here. We then extracted the amyloid from these vessels, shown here. We performed a ELISA analysis. And again, we found about 95% of the A beta accumulating in these vessels is the shorter A beta 40 isoform. We then looked in these models for cerebral micro-hemorrhages. Indeed, we found that in the case of RTGDI rats, we find cerebral micro-hemorrhages. These are visualized here by staining for perivascular hemosiderin, shown in blue. Here's again another small microvessel here that you can again see this perivascular hemosiderin staining. indicative of micro-hemorrhages. Similarly, we find micro-hemorrhages occurring in RTGD rats. Here's a surface meningeal vessel, and here a vessel within the thalamic area, again, where we see this perivascular hemosiderin. So both models develop cerebral micro-hemorrhages. We also found that in the case of RTGDI rats, we find there are capillary occlusions of vessels. These are occluded vessels that are actually calcified. They're stained with calcium here in black. You can again see these small vessels and capillaries here that are occluded. Similarly, in the RTGD rats, we find occluded vessels, again, stained with calcium. You can see these, occluded vessels. However, in contrast to these, which are occurring capillaries and small microvessels, we see this is occurring mainly at the level of arterioles. So different than vascular beds that are being affected here. So one of the things we wanted to look at too were some of the inflammatory responses that are occurring in response to this amyloid. So here we're looking at astrocytes. They're labeled in green with GFAP. Here we have vessels. This is an aged wild type rat. In red are the vessels. In green are the astrocytes. We see a normal distribution of astrocytes. In the case of the RTGDI rats, when there's amyloid on the vessels, which is blue, so on the capillaries here now, as I mentioned earlier, this is CA type 1, it's dysoric. It engages the surrounding parenchyma. We see a very, very strong astrocytic response. In contrast, when you look at the RTGDI rats, this amyloid is not dysoric. It's contained within the vessel wall, does not engage the surrounding brain parenchyma. And again, you see very little astrocytic response to this. We find a similar thing when we look at microglia. Here again, aged wild type rats. In this case, microglia are labeled with an antibody to IBA1. You can see that they're microglia around. They're in their resting surveillance mode. They have long extended processes. In the presence of this dysoric capillary CA type 1 amyloid, again, we see this dramatic increase in the number of microglia, and also the change from a surveillance mode to an activated state. When you look at the CA type 2 Dutch rats here, again, the amyloid is contained within the vessel wall, not engaging in surrounding parenchyma. Again, we see very little of a microglial response. They're mainly here in their resting surveillance mode. So again, very, very different inflammatory responses depending on which type of CA we're looking at. So just to summarize some of these findings here, when you look at these two models, okay, the RTGDI, it's a model of capillary CA type 1. RTGD is a larger vessel CA type 2 model. Okay, they're composed both primarily of A beta 40. Both models develop cerebral microhemorrhages. Both models have small vessel occlusions. And although not shown here today, both models also develop progressive cognitive impairment. However, again, when you look at the perivascular neuroinflammatory response involving microglia and astrocytes, it's very robust in CA type 1, but there's much less, if any, activation in CA type 2 in Dutch rats. So I'd just like to just present very briefly here some utility of this model that we've been used to do some preclinical testing. This was a study that was done in conjunction with Allen Pharmaceuticals. They have their compound here, mivelsiran, which is an siRNA investigational drug being studied for the treatment of CAA and AD. And this is a drug that's targeting APP expression. Again, the idea of trying to lower APP expression in the disease. So the study was designed, these are RTGDI rats we used here. They start, again, showing pathology about 3 months of age. As I showed, it progresses dramatically after 12 months of age. So in this study, we administered a single dose of their APP-targeted lowering siRNA, or just artificial CSF. After 1 month, the subset of these animals were collected to look at the levels of APP mRNA expression, and the remainder of the animals aged out for an additional 6 months, out to 9 months, where then we collected their brains and performed histology looking for amyloid accumulation and microbleeds. So here is just the results looking 1 month post-dose with the siRNA and looking at APP mRNA levels. And here you can see compared to the artificial CSF treated animals, there's about a 60% reduction in the expression of human APP mRNA in these rats. Their work has shown that this will actually extend out to 9 months to a year from a single dose. We then looked at vascular amyloid deposition. On the left, again, stained with thioflavin S to see the capillary CAA, as I showed in earlier slides. The artificial CSF control, we see, again, extensive amounts of capillary amyloid. In the case of the APP siRNA lowering treatment here, we see that the amount of vascular amyloid is decreased dramatically. We quantitated that in a number of these rats here, and we can see there's about an 80% reduction in amyloid accumulation on capillaries in these animals. We then looked at the micro-hemorrhages in these animals. Here are the CSF control rats. Again, we see the presence of perivascular hemosiderin here, indicative of micro-hemorrhages. This was greatly reduced in the APP lowering siRNA treatment. Here we looked at actually the occurrence of cerebral microbleeds. There was actually about a 70% reduction in microbleeds in the APP siRNA lowering treatment. We also looked at the size of the microbleeds and found similarly there's about a 70% reduction in the size of them. So this actually showed that this treatment here is very effective at lowering APP expression, thereby reducing the production of a beta peptide. This actually translated to much lower levels of CAA and a reduction in microbleeds in these animals. So we were happy that this study was successful, was part of the basis for them actually now going on to a clinical trial in CAA patients for looking at this treatment. So that's one of the examples we've been using these transgenic rats for to model for preclinical testing. Again, as I just showed you briefly here, the Alanam study where we used their APP targeted siRNA. We're also currently looking at A-beta immunotherapy amyloid-related imaging abnormality effects. We're doing that in combination with Lilly to try to understand why patients that have CAA are more prone to these effects. We also have trials going on right now. We're testing anti-inflammatory treatments for reducing CAA-related inflammation. And lastly, we're also looking at lifestyle factors and the impact of diet and exercise on the emergence, progression, and severity of CAA. So these are some examples of the utility that we're using these rat models for going forward. So for the final part of my talk here, I'd like to describe some work that actually was spearheaded by Joe Schrader, who was a postdoc in my lab and now is a research assistant professor here in the Department of Biomedical and Pharmaceutical Sciences, as well as the Ryan Institute for Neuroscience. So with these were studies here, we performed proteomic analysis on both of these rat models of CAA, trying to gain some insight into the molecular pathogenesis of disease. So again, I just bring the slide I showed up earlier again, that again, to emphasize, these are two very different vascular beds, capillary CA type 1 here, larger vessel CA type 2 here, some of the common features that they have, and also some of the strikingly different features they have, particularly with regards to inflammation. Excuse me. So first, what we did was we wanted to select rats to do this proteomic analysis. And what we decided to do was to select rats that have similar levels of A-beta accumulating in their brain. So this is ELISA analysis here for A-beta 42 and RTGDI rats, A-beta 40. And similarly, A-beta 42 here in RTGD rats and A-beta 40. So therefore, these rats from both models have similar amounts of A-beta accumulating in their brain, but they're accumulating in two very different compartments. The RTGDI rats, it's accumulating on capillaries, whereas the RTGD rats, it's accumulating in the larger vessels. So mass spec analysis was performed on a number of these rats. And here are some results. This is a volcano plot from RTGDI rats. In looking at wild type rats, RTGDI and RTGD rats, we were able to identify about 4,500 proteins per each rat. And what we did here in this volcano plot was to set arbitrary cutoffs. We were looking at proteins that were at levels at least 50% higher than wild type and with a significance value of p less than 0.05. So those are the ones that were identified here that were upregulated in red. There were 322 identified, and downregulated in blue here, which we identified 142. We did that same analysis with the same exclusion and inclusion criteria with the RGGD rats. We found much lower numbers of increased proteins, in this case 78, and decreased proteins 35. This is probably, to us, was not too surprising because this model here, there's a very, very strong perivascular inflammatory response and this surrounding parenchymal engagement. So there's a lot more pathology going on at that level in here, which probably explains why there are many more proteins that are up and down regulated. So what we wanted to do is, okay, we've looked at the upregulated proteins here, and we wanted to understand which proteins are unique to each form of CAA and which proteins do they share in common. And that's illustrated here in this Venn diagram. So we see from this that there are 36 proteins that are shared that were upregulated based on these criteria here between the RTGDI CA type 1 and the RTGD CA type 2 rats. And we can see there are a number of proteins that were unique here for the RTGDI as well as some for the RTGD. So this is just a summary, a brief summary heat map here of some of these selected proteins from each of these categories. Here are RTGDI specific proteins. We can see that we're upregulated or at higher levels because if you see very little change of them in RTGD rats. These are some proteins of these 36 here that were shared between both models. And then here are some proteins that were upregulated specifically in RTGD rats and not in RTGDI rats. So again, this tells us again that there are common and unique signatures for each of these forms of CAA. And one protein in particular here, which I'll discuss that we were interested in pursuing, is this protein here, HGRE1, which is upregulated in both forms of CAA, the CA type 1 and CA type 2. Joseph then took this data and performed some pathway analysis to get an idea about pathways that may be upregulated in each of these forms of CAA. And some of those are listed here. Here's an example of looking at proliferation of neuroglia. We see that, again, a score of 2 is considered significant activation. That's significantly activated in RTGDI, but not the RTGD. If you remember back on one of the previous slides I showed you, the RTGDI rats have a very strong perivascular glial response, whereas RTGD don't. So this is actually very consistent with this pathway. Similar, we see some of these other responses too. If you look at complement cascade, that appears to be somewhat activated in both. And this acute phase response, again, is elevated, but not significantly here in the RTGD rats. But again, this is an acute phase inflammatory response. We see that elevated here in RTGDI. So again, these are consistent with a lot of some of the pathology that we've characterized in each of these models. So I'd like to just finish up with talking about this protein HGRE1 that we found was elevated in both models of CA type 1 and type 2. So what is HGRE1? It stands for high temperature requirement protein A1. It is a proteolytic enzyme. HGRE1 regulates TGF-beta1 signaling and extracellular matrix proteins. Now, what's interesting to us is that this protein is associated with disease known as cerebral autosomal recessive arteriopathy with subcortical infarcts and leukoencephalopathy, otherwise known as carousel. This is a cerebral small vessel disease. And patients that have carousel, they have it because it results from having one of multiple mutations in the proteinase HGRE1 that disables its proteolytic activity. So carousel patients have a proteolytically inactive form of HGRE1. It's been hypothesized that sequestration of HGRE1 in CAA reduces its proteolytic activity. So that may be a commonality between these small vessel diseases is a decrease in the amount of active HGRE1 protein. So we wanted to look at HTRE1 in our different models here. These are RTGDI rats and wild-type rats here that were, again, labeled with, stained with thioflavin S to identify fibrillar capillary amyloid. In the wild-type animals, you do not see any, as you'd expect. We see a very low background staining of HTRE1 in red. When you look here, when you have capillary amyloid shown in green, we see this dramatic increase in the amount of HTRE1 staining. We found a similar thing in RTGD rats. Here's actually the measurement of the protein increase that we see in RTGD rats. And when we look at vessels here, these are RTGD rats, these are HMH wild types here. These were stained again with thioflavin S to see the vascular amyloid. You can see this vessel here, decorated vascular amyloid. We don't see any vascular amyloid staining in the vessel here. You look at HTRE1 labeling, you see it strongly colocalizes with the vessel, with the vascular amyloid. Again, we see really no vascular, very little, if any, HTRE1 staining here. So in both of these diseases, we see an increase of HTRE1 labeling associated with vascular amyloid. So since HTRE1 is involved with regulating TGF-beta1 signaling, we looked at that from our proteomics data. And here's a condensed heat map looking at a number of proteins that are associated with TGF-beta1 signaling and showing that they're markedly increased here. We then went on and for the ELISA analysis, looking at TGF-beta1 protein in the brains of these rats, and found indeed a significant increase of TGF-beta1. We found a similar thing when we looked at heat maps of TGF-beta1 signaling proteins here in RTGD rats. And similarly, we find this increase in TGF-beta1 activity, protein by ELISA. So there appears again to be a strong connection between HTRE1 increases and TGF-beta1 signaling and the TGF-beta1 levels in these two different models of CAA, again, which has shares some commonality with that disease carousel that I mentioned earlier. So we're very interested in following up on this and pursuing this link between HTRE1 and TGF-beta1 and its role in pathogenesis CAA. So to just summarize what I shared with you today, we have generated these RTGDI rats in our model of capillary CA type 1 and has a strong CA-related inflammation. We've also generated RTGD rats that are model of large vessel CA type 2. They present with a much more muted CA-related inflammation, again, in the absence of any disordered vascular amyloid. The RTGDI rats and RTD rats, again, they have some common and distinct pathologies associated with their respective types of CAA. And both rats models have common and distinct changes in their respective cerebral proteomes. And this also reflects a stronger perivascular inflammation in RTGDI rats. And lastly, the RTGDI and RTD rat models share elevated levels of cerebrovascular amyloid-associated HTRE1 and increased levels of TGF-beta1. And this is something that we're pursuing to understand the role of this in the pathogenesis of the disease. And finally, I'd just like to acknowledge people in our lab done this work, Judy Ann Davis, who spearheaded the generation of these transgenic rat models, Bethany and Mark, who maintain these colonies and done all the behavioral analysis, Dr. Xu and Dr. Zhu, who have done all the detailed quantitative pathology, and finally, Joe Schrader, who's really spearheading the proteomic analysis of these different models. So I will stop there, and I thank you for your attention, and I'll take any questions.
Well, great. Thank you so much for this wonderfully comprehensive and insightful overview of this devastating, disease. I encourage folks in the audience to enter questions into the chat, and we can relay them. Meanwhile, maybe I could ask, what is like the standard of care for these patients with CAA?
So it's difficult. So currently, it's difficult. You cannot identify outside of mutations. If you just look at sporadic CAA or Alzheimer's disease, the current clinical diagnosis is based largely on imaging, neuroimaging studies. And in that case, patients typically have a very severe pathology at that point. where they're developing microbleeds and hemorrhages. So it's difficult actually to identify earlier patients, but patients they do identify that based on the neuroimaging criteria for being probable CAA. There's not, I mean, they really just try to control vascular effect. They try to control blood pressure, things like that. There have been some studies, some patients that have very strong inflammatory reactions to CAA. Not all patients do, as I showed you, it depends on what type of CAA you have. But if there is a strong inflammatory response, there have been studies that show, again, that treating with strong anti-inflammatory reagents can actually reduce that inflammation and reduce any further bleeding or hemorrhages from occurring. So that's basically what's available for patients that present at least with that clinical sign of having bleeding. But besides that, at this point, it's really controlling just vascular factors such as hypertension and things like that. Again, now, as I showed, this one drug is in clinical trial now for CAA. We'll see where that goes. And so there are other things being looked at as well as some other more specific anti-inflammatory agents and things. So that's currently where it's at.
Yeah, thank you. Following up on that, what any numbers with regards to prevalence of this in the population?
Well, as I said, so there've been studies that have shown that for individuals that are over 80 years of age, that's estimated at least 50% of them have some amount of CAA. I mean, some not a lot, maybe, but they find it in at least 50% at some level. So it's quite prevalent in the elderly population, even in the absence of Alzheimer's disease.
So there is a question in the chat. Do these rat models accurately represent human CAA, or are there limitations?
I'm the first to acknowledge there are always limitations in these models. I mean, they are rodents for sure. So there's clearly that difference there. But they do reflect many of the features that you do see in CAA. So I think there's some good utility for them. But again, the caveat always is in this case, one is that they're rats. The second thing, major caveat that I have with them, they are transgenic. And so one of the shortcomings that I see with transgenic models is you're expressing a protein in a given cell type. In this case, it's in neurons. You're expressing human APP as a transgene in those cells to make this human A beta peptide that accumulates in the vessels. It gives you the pathology. But the question I always have in my mind is it presenting that pathology or developing it in an accurate way? And I think that there really are some shortcomings for that by using transgenics. So that's why we've actually moved on to some next generation models where we've done gene editing. And in this case, what we have done is these two transgenic models here, we have now recently turned into gene edited rat models, where we've gone into the endogenous rat APP gene, and we went specifically into the A beta region. And it's different from humans by 3 amino acids. So the first thing we did was to change that, so it's now human A beta. We then engineered in either the Dutch or the Dutch IO mutations with the idea now that's producing human A beta with these mutations in it, but it's occurring through the endogenous rat APP protein. And this way, it's being expressed in all the right cell types, in all the right places, and at all the right time. We think these are much more physiologically relevant models going forward to study for testing things if you're trying to intervene in that process. So that's, I think, an advance that we're trying to do now. I think they'll even improve upon these transgenic models. So again, with transgenics, there are a number of caveats always come with them. So we certainly have to recognize those when we're trying to translate what we find here to what we find with humans.
Right. Yeah, the gene editing approach is should offer a number of advantages. That's great.
Yeah, that's why we've moved on that direction.
Yeah. What's known about the sort of life cycle of the mutated proteins in terms of CSF, transport, release, blood vessel? Are there any biomarkers in the blood that are?
So we have a study actually that we just completed, and it's ongoing now too, where we've been looking for CSF biomarkers using our models, and we are working with colleagues in the Netherlands who have a big CA data bank there that we looked. So we've been attacking it from the human side, us from the model side, and we've been communicating, trying to see what do they find, do we find out in a model, and do we identify any things that come out in the models that we can translate and then go look specifically in human CSF and see if those proteins are changed. So that work's ongoing right now. We have a number of candidates that we're looking at. With regards to A beta in the CSF, so and this has actually been, we've demonstrated this in our rat models, and it's also been shown in humans. When you look at CSF levels of A beta, the A beta 40 peptide drops dramatically in CSF with the presence of CAA. And that's hypothesized because that's the form of A beta that's accumulating in the vessels. So as it accumulates in the vessels, there's less in interstitial fluid in the CSF compartment. And we have shown, we've been able to do this. One of the advantages of the rats is we can do time studies. And with the humans, they have a CSF sample and they have it. We've been able to show and document that as the pathology develops and becomes more and more severe in the rat model, the levels of A beta 40 in the CSF drop in parallel with that.
Another question that comes to mind is whether there's any accumulation or effects in the peripheral
No,
vascular system.
there really has not been, this really appears to be a CNS disorder. It seems that the cerebral blood vessels were affected. Even in the familial patients, there really hasn't been any indication that you find any peripheral vascular pathology or accumulation.
Right. Okay, it doesn't look like there are any other questions in the chat. So want to thank you, Dr. Van Nostrand, for a terrific talk. We really appreciate your work, and wish you good luck and that as the work proceeds, and especially with the exciting advances with the gene editing and a rat model. So I want to thank our audience for their participation and engagement, and ask you to complete the survey that will be distributed to you. And I wanted to let everyone know that our next CTR seminar will be on March 13th, and Dr. Mary Carskadden will be speaking on biobehavioral underpinnings of teen sleep. So we look forward to that presentation. So thank you all, and have a good rest of the day. Appreciate it.
Thank you. Thank you for your attention today.
Okay.